Okay, welcome everyone to the second David Finney Centenary Lecture. These lectures were initiated by the newly established Centre for Statistics in the University of Edinburgh, with the first one of last year. And they were to honour David Finney, who was the first professor of statistics in the University of Edinburgh when he joined in 1966 and retired in 1984. And he celebrated his 100th birthday in January last year. During his time in statistics, he was both president of the Royal Statistical Society and also the International Biometric Society. And now these annual David Finney lectures are co-sponsored by the Centre for Statistics and also the Royal Statistical Society Edinburgh Local Group. And it's our great honour this year to have Professor David Dunson give the lecture. So he is the Arts and Sciences Distinguished Professor of Statistical Science, Mathematics and Electrical and Computer Engineering at Duke University in the States. His visit to Scotland is being supported by the Carnegie Trust, and he's one of only 2 Carnegie professors for this year. David is an eminent statistician, probably known to many of you in the room. He has a global reputation, so it's great to have him in Scotland for this time. He's a fellow of the American Statistical Association and also the Institute of Mathematical Statistics, and has won numerous awards during his career to date, and I'm sure will continue. So I'll pass over now to David, who's going to give our David Finney lecture.
Okay, thanks a lot. And please interrupt me as we go. I'd like to keep this as informal as possible. And let's see if my slides come up. Okay. So today I wanted to talk about, I'm a statistician and data scientist, but I've been involved a lot lately in neuroscience studies. And so I wanted to focus in particular on studies of the human brain connectome and kind of get into some of the details without going into any of the technical details, looking at relationships in human brain structure and in other traits of the people, like are they have substance use or what's the level of education, et cetera. I'll get into the details. Okay, so I'll start out talking about brain connectomes. And so what is a brain connectome? How do we measure it? Who cares about a brain connectome? What does it mean? So I'll go kind of slowly through that, interrupt me with questions as we go. The brain connectome is a kind of type of network, and so I'll give a little bit of background on network analysis, which has been a huge area of data science in recent years, and often people are thinking about maybe a social network like people on Facebook or employees of a company or something like that, and it's interesting that you can use similar data science tools as people use in social network analysis to analyze the brain structure and its relation to traits, okay? And then I'll kind of loop back and talk about that a little bit, and then I'll focus on some really pretty amazing results we have for some of the data and applying these methods. Okay, so what are we interested in? And so really we're interested in understanding the function of the human brain and how it varies across different people, and how that variance is related to traits of the people, okay? And so I think it's really remarkable how little we understand about the human brain relative to other things. And so medical science has improved incredibly in the past decades. But the human brain has remained relatively elusive, particularly neuropsychiatric conditions, like do we really understand the sources of depression and anxiety and schizophrenia, other things like mishaps that happen in the brain, okay? So the understanding is really limited, I'd say. And so the lack of understanding about the functioning of the human brain and its relationship to traits of people has really had broad sweeping practical ramifications. And so the first I would say is that there's been quite poor diagnosis and treatment of mental health diseases and disorders. And so there's these criteria where we're diagnosing people based on symptoms, and the symptoms are very coarse. And so if you look at symptoms of mental health conditions, and then you kind of put it into this DSM criteria, then what happens is that if you talk to neuroscientists, what happens is that a given symptom or set of symptoms is not clearly defining an actual disease for the person. There's what's called endophenotypes, and so there's actually something that goes wrong in the brain. If something goes wrong in the brain wiring, that can cause many different types of conditions, okay? So that's the first problem. The second problem is that if you have a particular symptom, there can be many different causes of that symptom, okay? And so it becomes very difficult to pull things apart just based on looking at symptoms. And so it's necessary, I think, to really try to peer inside the brain, get some better data on the brain to improve things like diagnosis and treatment of mental health disorders, which is now quite archaic. Another example is sort of one-size-fits-all education programs. So if people's brains are wired to learn in different ways, then we should be adapting education to take that into account. Okay, so if you're wired to learn more visually, then maybe we can pick that up by looking at your brain, then we should be giving you a different type of education program. Okay? There's a lot of uncertainty in what factors diminish or improve cognitive functioning, and so you can imagine that your brain, how healthy it is super important to your well-being, but we know massively less about that than how we can train, your body to do well in sports or be healthy or something. Like you go to the gym, you lift weights, you might go running, you're swimming or something, and then you can have this very noticeable effect on your cardiovascular system and your muscular, et cetera. But it's not really known so much what's impacting your kind of brain functioning nearly as well, I think, even though there's a close analogy there. So what's our goal is to essentially use modern technology, and particularly image technology and data science technology, to kind of transform this landscape. And so we want better tools to be able to take somebody who's maybe they're healthy, maybe they have some conditions they'd like to understand, and you go in and you can maybe put them in a scanner that non-invasively and get some information on their brain that can tell us about that individual's traits and the reason for different conditions that they might have. It might also explain how that person learns in an educational program, for example. Okay, so that's the kind of thing we're interested in. Okay, so what's been the big problem in the area is your brain is encased in this skull, and we can't see into it. It's sort of obvious if you have cardiovascular condition, we can get quite good information on the heart, for example, or if you do an exercise program, you can see gains in how strong you are and in your muscle fitness, et cetera. But in the brain, it's all sort of hidden from us, okay? And then if we go in there on a fine scale, there's an absurdly huge number of neuron cells in the brain. And so here's kind of a picture of these neurons. And so in your brain, there might be 100 billion neurons, okay? And they're all interconnected. And so you have these kind of structures that are all interconnected in the brain, and it becomes incredibly complicated, okay? And so you could think that these neurons are almost like some sort of electrical system controlling the wiring in your brain and information processing. And one of our grants right now, there's a new type of technology where you can take an animal, and the animal's alive, and you're actually kind of cutting off a little piece of the skull, and you can take video recordings of the neurons firing. And what we're interested in doing is to try to figure out how information processing is occurring. So why are people so smart? We have all these neurons, but they can't just be firing randomly. They have to be doing some sort of distributed information processing. And so if you work on computer science and data science and you have problems where you're trying to do machine learning, we know that if we have a big problem, we need to break it up into smaller problems, and we need to distribute that information processing across maybe different computers, for example. And so the brain has to be doing something like that. It has to be distributing information processing across different locations in the brain. but it's really not well understood how that's happening. And so we've been doing mouse studies where we're kind of looking into that using optical imaging, where you can actually take videos of these kind of neurons firing. Okay, so the neurons are all connected in a big giant network, but we can't really see these individual neurons in a person. Okay, that's impossible. So you're particularly a live person, your brain's in your skull, and then we only have some sort of indirect imaging technology to kind of try to indirectly, certainly not at the resolution of individual neurons, get data on the brain. So one thing that's really important is that the neurons are not really randomly located or sporadically throughout the brain, but they're located in a really intricate spatial pattern. And I kind of like to draw on the board a little bit. Where did I put my hand? And so you can imagine that, there's these individual, is that a chalkboard? And so see, what's actually happening in the brain is there's these larger, what are called, I'll talk about this, fiber tracts, fibers or something, okay? And so these fibers are actually of a size that we can kind of get data on. And so within the fibers, there's all these like a huge number of neurons that are all connected. So if we look at these guys, they're all they're not like just going everywhere in the brain. They're actually tightly connected in these sort of wires, these kind of coarse wires, and the fibers are kind of little filaments. The neurons are kind of tiny little filaments that are kind of spatially aligned in these big wires, okay? And if you look at the brain, there's actually kind of big highways, and there's smaller back roads, and it's this sort of a traffic system or network in the brain, okay? And so we can't pick up on these individual neurons, certainly not in the human brain, but we can kind of get data on the location of these kind of highways or back roads in the brain and where they are spatially. Okay, and so in particular, the brain's broken up into gray and white matter, and these fiber tracts are through the white matter of the brain, okay? And so you could call this a fiber tract or a fiber bundle, the sort of bundled together neurons. Okay, so there's been a lot of incredible advances in brain connectomics. So I was at there's a Howard Hughes Medical Institute in the US, which is a very famous place, and at Genelia Farm, which is one of their institutes, they work a lot on neuroscience. And one thing that they're trying to do is kind of map the fly brain. Okay, and so I'm just trying to give you a scale here of like a fly brain versus a mouse versus a human brain to give you an idea, but it's been a ridiculously hard task to map the fly brain. And what do I mean by mapping the fly brain is we can take the fly brain, and now there's this amazing technology where we can slice it into these kind of pieces. And each piece is on the scale of nanometers, okay, some number of nanometers. So that's incredibly tiny little slices. And then we can image each slice, and then each slice, what it'll look at, it'll look like something like this. It'll be like a slice through the neurons. And then I get another slice, and I'm trying to line up all the neurons and all the different cell bodies from each of those slices. And I get an enormous stack, this giant stack of these slices, even though the fly brain is so small relative to the human brain. And then if I've done that successfully, which has been really, really difficult, then I have a whole map of all of the neuron interconnections in the fly brain, and I can try to look at how brains are structured. Because it's pretty remarkable in biology. You can see that even a very simple animal system like a fly is much more similar to a human than you would think they would be. Okay, and so then we can, on the fly, we can do these nanometer slices, and we can do what's called EM connectomics and try to map the whole fly's brain. Okay. On the mouse, we can do things like I was talking about with this kind of new technology where we can do what's called calcium imaging, where we can get these amazing videos of individuals' neurons firing while the mouse is doing things. So the mouse has to be still, and so you actually put the mouse in this sort of virtual reality machine. And so the mouse thinks it's doing things, but it's actually completely still. Yeah. It's kind of cool, or gruesome, depending on how you look at it. And so for humans, we can't really do that. There has been some studies in monkeys where they'll actually try to look at relationships between indirect measurements we get of these kind of fiber tracts and the actual true fiber tracts that we get by dissecting the brain. Okay. So the problem is that we have our brains are like encased by our skull, and the skull is blocking information processing. And we had a previous grant which was really quite interesting on brain-computer interfaces, funded by the U.S. Defense Advanced Research Project Agency. And so the problem they were interested in, which is kind of scary in some sense, was there has been these experiments where you have a monkey, something like that, and so you could insert an electrode array in the brain of the monkey. And we're doing these studies right now in mice as well, and rats. But you insert an electrode array in the brain of the monkey, and then you could tie the monkey's arm down and have the monkey control a robot arm. And the monkey can then do things with the robot arm. It can like peel a banana, eat a banana, and manipulate its environment. Just based on, so it's this kind of decoding problem. So the electrodes are recording those neurons spiking the electrical activity of the individual neurons and the local field potentials, which are locally averaged electric activity, and it can use that to manipulate the robot arm. And interestingly, even if there's a bias in collecting the data, the monkey can figure it out. And so initially the monkey's arm will be kind of erratically, it'll miss the banana, but then after a little bit of time, it'll kind of be able to figure out how the decoding is working in the device, and it'll be able to pick up the banana and manipulate its environment. So it's pretty amazing, really. Yeah, and so what the problem was, so the thought was, well, maybe we could do that with humans. Like, what if a human soldier, and it's a defense grant, they had their arm blown off or something? Well, certainly they want to still be able to assemble their rifle and shoot people still, and so maybe we could have them have a robot arm or something. And so you could do that in people, as you did in monkeys. That's certainly possible. But what happens is that as you put these electrodes in, and so what does the body think? Well, that's a wound, okay? So the brain is wounded, and so what happens is that these special cells, it's amazing how plastic the brain is one thing we learned from this, that these special cells called microglia cells will rush in very amazingly quickly, and the cells will get all around that electrode, these microglia cells, and then in an adult human, like very quickly these cells migrate in there. And what happens is then the quality of the recording of the brain is diminishing as that wound gets healed by all those microglia cells coming in and encasing the electrodes. And so what people are trying to figure out is there a way we can kind of turn off that wound healing response? And so that we can kind of keep people, and if we did that, then it would seem like you can have people's brains automatically controlling devices. Like you could have, your brain could control a drone or remotely control an airplane or something like that. So certainly that's possible, but there's this major problem where the wound healing is kind of making the signal worse over time. Yes, you get it in the monkeys too, yeah, definitely. Okay, let's go on. And so one of the studies, before I get too into the connectomics, we do a lot of these rodent studies, and so you can actually design a rodent. Like now we have wireless devices, and so you put a device on the top of their head, and then you could design experiments in the animals, and the animal might be actually designed, genetically engineered to mimic schizophrenia or mimic autism. And so then you have a normal type animal and an animal that has a gene knocked out that's mimicking autism, okay? And so you have both of these animals, and then since we have the wireless device, we can put them in an environment. And what we're doing now is you'll put them in an environment where there's a rock in the cage or another animal and then you can be recording their brain activity as they're engaging in social activity, either with an inanimate object like a rock or another animal, and you can see how that brain activity is different between the autistic mouse and the non-autistic mouse, and then start wiring. What you'd like to learn is sort of brain subnetworks that are indicative of disease, okay? And so, well, here's some brain subnetworks. It tends to be when you're undergoing social interaction for an autistic individual, they tend to have a particular set of brain regions are lighting up together in that individual, or they tend to not light up as much in that individual. And so those are brain subnetworks that we're trying to identify. Okay, I'm not getting very far in my slides here, though. Okay, I said all that. Okay, so human brain connectomes, which is the main point of the talk, is how do we define these? And so, in some sense, it's the set of interconnections of neurons in your brain, but we can't collect the data on the level of individual neurons. And so this just shows a kind of image of one of these fiber tracts, okay, these white matter fiber tracts. And so within these tracts, there's a ton of individual neurons that are spiking, and the kind of flow of electricity through these neurons is kind of going along these tracts. And these tracts are connecting different regions of the brain. And so that's controlling kind of whether a certain region of the brain is activating, and also the interconnections or communications among regions of the brain is kind of controlled by these kind of tracts, okay? And along the tracts, we have a diffusion of water molecules, okay? And so a water molecule will diffuse along these tracts. In gray matter, it just diffuses weakly in all directions, but it diffuses directionally along the white matter tracts. And that's the only information we're going to have about where these tracts are that we're trying to pick up by an MRI machine, okay? Yeah, so we'd like to figure out relationships between the brain structure and human traits. And so in order to do this effectively, we're going to need to have like a medical study or an epidemiologic study where we enroll maybe thousands of people, hundreds of thousands of people. For each of those people, we measure their brain structure, and we also measure stuff about them. Maybe initially we could have a baseline study of healthy people and see how brain structure is related to traits of healthy people, like different measures of IQ, maybe their lifestyle, maybe some demographic factors, maybe some educational factors, et cetera. And then after that, we could maybe look at studies particularly targeted towards depression or Alzheimer's disease, et cetera. But we really need an inexpensive way to non-invasively measure the brain connectome for each of the people in some big study in order to figure this out. And it has to be done accurately enough. Like you can imagine there's these fibers. If we have some image of the brain that's not at fine enough resolution, we might not be able to figure out that this is a separate fiber than some other thing that's kind of crossing over. We might put those together and then get a crappy estimate of the brain connectome. So how I'm going to define the connectome is it's the interconnected network of white matter fiber tracts, which are sort of highways for neural activity and communication. And I'll show a bunch of cool pictures. Here's one of them. Okay, so here's one person's brain connectome, okay, which is recreated by various types of fancy MRI technology. And so if I kind of zoomed in onto this, you can kind of maybe see from where you are little hairs or something. Like each one of those is a little fiber tract. And so in a normal human brain, there'd be about 1 million white matter fiber tracts. And you can see that they have this kind of structure. Well, these are the two hemispheres of the brain. So there doesn't tend to be that many connections across hemispheres in the human brain. There's many more connections within a hemisphere of the brain. And also there's a lot of kind of spatial geometric structure. You know, of course there's kind of local, things tend to be more connected locally, but there's also long-range connections across sort of major white matter fiber highways, essentially, okay? And so you can imagine that, well, this is one person's brain. Another person's brain who might be better at mathematics might have a little bit different picture. and another person's brain is like amazing at art or something or music, they might have a little different picture. And we'd like to figure out those relationships. Okay? So this is just one person. And so we did this using a type of magnetic resonance imaging technology. And one of the big advances in recent years that's kind of enabled some of the results I'll show today is basically bigger magnets in the MRI machine so that we can resolve these fiber tracts at a finer resolution. Before the last couple of years, you just couldn't do that, and basically the data were a bunch of measurement error and noise, and then you couldn't find any interesting relationships. So the data are very indirect. We don't directly measure where these things are. We're only indirectly measuring it through diffusion of water molecules in the brain. Okay, so here's some other pictures of, this is again one individual's brain. Okay, and so this kind of shows from different angles their brain connectome. Okay, so it's pretty cool. So you kind of see again these kind of feathers or hair. Each of these are little white matter fibers, but then they're arranged in this intricate kind of geometric pattern. And there's been a lot of concern about what you call measurement errors in reconstructing the brain connectome. And so we get a picture like this, but for nobody do we have their actual locations of these fiber tracts. We're only indirectly estimating the locations of these tracts based on a kind of statistical approach where we get indirect measurements of these diffusion of water molecules. Okay? Maybe they're not real, they're just some artifact of the computer processing. Okay? And so recently we just had a paper come out which really I think improved the processing a lot. So taking the original data on water diffusion and then turning it into one of these kind of cool pictures in a way that has a lot of signal. Like we still don't know that it's really an individual's true brain connectome, but it's super related to a lot of different things that are interesting, and it's reproducible. And so if I do this today on a given person, like if I take a whole bunch of people and I use our processing separately on different scans from the same person, and so I take a scan on me 5 times, and I'll take a scan on you 10 times, and then I'll do that for everyone in the room, and then I'll just throw all those scans in a big bag, and then I'll just cluster those scans to see which ones are most similar. The clusters are going to correspond very, very closely. They're going to be the individual labels, and so you're going to be able to, in an unsupervised way, figure out which individual's brain scans go together. And so that's strongly suggestive, at least, that it's reproducible. It's also, you can also distinguish between different individuals' traits based on their brain structure. And so that's suggestive that the measurement error is not such a giant issue. Okay, so here's zooming in some. This is kind of cool. So it's hard to see the whole brain connectome at once because you have like a million narrow little tubes snaking through 3-dimensional space, and it's just like a bunch of colors. But if we zoom in, this is for 4 different people looking at the same regions of interest of the brain. And so these are particular regions that have a known biologic function, a pair of regions. And so then now the green curves those are fiber tracts connecting that pair of regions. And so you can see that there's kind of similarities across people, but there's also a lot of differences. And so this individual, these people, well, this guy or girl has just a bunch of, and then the kind of highway right here, and then there's maybe just a couple of other fibers. And this one has a couple of 3 highways, and then this one has this crazy route kind of going over here. And so maybe there's something different about this person because they have this like very different track going over there between the same brain regions. And so can we kind of figure that out somehow? And so, yeah, we can run large studies now. And what we're going to be analyzing, there's this amazing resource, the UK Biobank. We've been looking more at this kind of US study, the Human Connectome Project, and I'll show data from that today. So we have brain imaging data for over 1,000 healthy adults between 22 and 37. So they're kind of young, healthy people, so like a baseline study to figure out what's going on. There's also really rich information on what I'll call traits of the individual, which include cognitive traits, you could imagine giving person in the study something like a visual spatial processing IQ test or something, or IQ tests in different domains. And I'm really good at doing visual spatial processing or another type of task. Then I'll get a high score. Maybe that also means I have a particular type of brain structure. Okay? Sensory, emotional, educational, substance use is one of the most interesting findings we had, and I'll show that later on in terms of alcohol and marijuana use having an adverse effect on the brain. Apparently adverse effect on the brain. It's an observational study. Okay, and so I should really give a lot of credit to my former postdoc, who's now at the faculty at University of Rochester, is sort of guru of processing these brain connectomes. He's fantastic. Okay, so what do we want to do? So we have a bunch of different brain connectomes, and we have different traits for many different people. That's the sort of data we're faced with. Okay? And our goals are to kind of develop data science tools for characterizing variation in brain connectomes across people. And so the first thing we noticed is, in doing some basic analyses, is actually people's brain connectomes are very similar. Like most of the structure is shared across people, even though there's definitely very distinct differences. Most of the structure is quite shared. Okay. Very similar brain connection structure across different people. And so we want to apply the tools to relate connectomes to human traits and exposures. And so if you have some adverse lifestyle factor like binge drinking, then we'd like to see what impact that has. And we'd like to produce results that are really easily interpretable. And so we don't want like a black box that says binge drinking is related to brain connection structure. We would like to say, well, what does it do to brain connection structure? And then maybe we can try to figure out why does it do that. Okay. And so one of the big challenges is one of the challenges of data science in general for biomedical research or for, in general, if we have high-dimensional data and we want to figure out relationships between that high-dimensional data. In this case, it's a brain connectome. It could be somebody's genome, or it could be some other measurement, very high-dimensional measurements you take of them from heart scans or something. So if we have a lot of like high-dimensional complicated data, and then we don't have that many people to study, then there's a very big problem there in trying to figure out what's going on. So we need some type of dimension reduction to make sense of the data. And so that's where this sort of network analysis comes in. And so network analysis has been a really, really hot area. You can imagine like Facebook and all these things. There's all these social networks, and people are interested in studying them as kind of sociologically. And also data scientists are interested in studying the properties of networks. So before I go into some of the details and show the results, I'd just like to note that this lecture is in honor of David Finney, who is a famous statistician who worked at the University of Edinburgh for many years. But one thing I found really interesting is that he had a lot of impact on developing systems monitoring drug safety and had a big impact on statistical methods for dose response and risk, okay, which is something I worked on a lot early in my career, and so I'd actually been influenced by him. But in these kind of cases that he was working on, you could imagine that you have some dose of a chemical. This is the weirdest board ever. You have some dose of a chemical, and then you have like risk. So you have some, as dose of a drug increases, then we have some risk of cancer going up, okay? So that's the kind of setting you're in, and then you want to be able to like set regulatory guidelines for where safe doses are, regulate the pharmaceutical industry, regulate environmental chemicals, et cetera. But you have this kind of setting. So in these kind of modern settings that we're kind of moving into, it's less and less common where we have this kind of risk of cancer. We have, people are complicated, health outcomes are complicated. In our case, we'd like to say, as like alcohol consumption increases, how does what change? How does your brain connectome change? And so you could think, well, maybe this is like the probability that we don't have an edge between particular regions of the brain, but there's many, many regions and the data are intricately connected. And so you have to kind of transition these simpler methods that these pioneers developed to these more complicated kind of object of data, functional data. Okay, so I'm really honored to be associated with his name in this lecture. And so kind of in that honor, I'd like to kind of look at kind of drug reactions on the brain as one of the examples that I'll get back to. So brain networks in relation to exposures. And so in Finney's work, again, he was interested in the impact on a 0-1 indicator of an adverse response. And either something bad happens or not, but we have the entire network of interconnections in the brain. And instead of looking at pharmaceuticals, which we don't have data on here, I'm going to be looking at sort of marijuana use and drinking too much alcohol as kind of an example. Okay, so we think what would be our hypothesis would be that potentially if you drank too much and it's having an adverse effect on your brain, maybe some of these connections kind of go away and your brain becomes less interconnected. And so we have a decrease in connectivity between certain regions of the brain that would seem to be bad. Maybe if you instead have some positive factor, like you have some education program, that might increase connectivity. And one of my collaborators runs studies like that, where he'll measure the brain connectome, and then he'll have the person go through some educational program. Like one was learning to juggle, and like so you learn to juggle over some amount of time, and you could actually pick up increases in connectivity and size of white matter tracts between certain regions of the brain after you learn to juggle, or after the students took like a linear algebra class or something. You could pick up some changes. It's kind of funny. So social networks, and so in recent years there's been a huge interest in analyzing social networks, and so this is most of the examples are kind of silly, but this is the Game of Thrones network. I don't know if Game of Thrones is a popular show over here, but it's very popular in the US, and so here's like some of the different characters, and so if you just look at take the data and then kind of cluster the data, look for communities in the data, then it kind of makes sense. If you know about the show, it'll kind of cluster them into these kind of communities. That makes sense. So that's one of the main things that people do in social network analysis, is they're looking to uncover communities and exploit that. So what is a network? A network is a set of nodes and links between nodes. And so in a social network, the nodes are people, okay? And then a link between different people, that might be because the people are friends or not or something. And so if you ask a bunch of people who your friends are, then from that you can kind of reconstruct a social network. In our case, we instead have a kind of a neural network. We have the brain, and then we have interconnections between regions of the brain. And so it's a little bit different, but the data structure is quite similar. So there's a lot of tools for analyzing these networks. Okay? And so one of the really useful classes of mathematical models, which I'll not be going into the technical details on, is what's called a latent attribute model. And so for social networks, you can imagine that, well, we're trying to have a model, like a mathematical model for predicting who's going to be friends with who, okay? So you could imagine, well, how could I write down a model? Well, I could say, well, there's some attributes of the person. It's like, well, I'm into swimming, and I like doing some math and neuroscience or something, and I like to hike or something. And so I have some latent attributes. I might not observe those directly for a person, and then somebody else might have a set of attributes. And then if your attributes are similar, then maybe you're more likely to form a connection. You might be more likely to be friends with that person. Maybe another of the attributes is actually where you're located spatially. Okay? And so these latent space models, which were kind of pioneered by, we recruited him to Duke, Peter Hoff, who's across the hall from me at Duke, are a really useful, flexible way for characterizing network data. So you can kind of do a good job flexibly characterizing network data. And so for the brain, we could do the same thing. We could think, well, each region of the brain has some latent attributes, and that latent attributes for different regions of the brain, they're kind of controlling how connected the brain is. Okay? And then we could allow those latent attributes to vary systematically across different people, and then we have some sort of very flexible model for characterizing differences among people in their brain structure. And so that's sort of what we're doing. And here I'll show some data. So this is the caption's too small to read. I'm sorry about that, but we had a study where we were measuring creative reasoning scores, and so this is a individual at a low creative reasoning score. Here's an individual with a high creative reasoning score. And so This is their brain structure. And so the node, those little circles, that's a given region of the brain. We have two hemispheres of the brain. There's an edge if there's at least one of these kind of fiber tubes connecting those two regions of the brain. So we're representing mathematically this more complicated object as a kind of network. It's a bunch of nodes, brain regions, and then edges are not between the nodes. And then we want to characterize how people vary in terms of their brain structure, okay? And so you can see, like, here's some different people. They vary a little bit. I mean, it's very similar, but they're varying. And, you know, maybe that variation relates to this label, whether they have a low or high creative reasoning score. And so we want to kind of estimate something like a population distribution of different brain structures. And so you can imagine, what's the population distribution? Well, if you measured like everyone's body weight, you know, it might just be some sort of bell-shaped curve, like Gaussian normal distribution. If you measured like a body weight for everyone in the population, or something like that, that's a distribution. So, but we want a distribution for like brain networks, and so you can't plot that easily on the board, but you can represent it mathematically with these kind of latent space models using a Bayesian approach. Okay, so I wanted to touch a bit on Bayes because partly because he studied at Edinburgh, and also because I do Bayesian statistics. And the Bayesian approach is really useful for a variety of reasons. And so, this is the Reverend Thomas Bayes, and there was a really interesting work by Stephen Stigler at University of Chicago in the States, who's a statistics historian. And so he was shedding doubt on whether this is actually a picture of Bayes or not. Certainly this is what everyone uses as a picture of Bayes, but it seemed like maybe it was actually his brother or some other Bayes that wasn't the Bayes, and so it's kind of funny. But the key with Bayes, and there was later on Laplace came up with a similar idea, was it's a way to characterize uncertainty in learning from data through probability distributions. And so what do we want to learn? Well, we want to learn a population distribution of brain networks and be able to kind of relate the brain network distribution to human traits. And so when we're doing that, there's going to be a lot of uncertainty. If we just got some one guess, best estimate of this population distribution based on the data we had, we might be pretty far off. And so it's really important to put uncertainty in there, and Bayes' rule provides a way to do that. So I think he was motivated partly by religious reasons, because he had this kind of conditional probability distribution, and maybe you could kind of go all the way back to the creator or something using this kind of sequence of probabilities. Okay, but in our case, we want to allow uncertainty for the population distribution of these kind of brain connectomes. And we'd like to allow that distribution P to change flexibly with traits of the individual. Okay, so let's kind of apply some of these methods and then see what happens. And so, so I have some really cool results for the Human Connectome project. I'll just first show some quick results for creative reasoning and Alzheimer's disease. And so what we had here is we did a study where we had different students at University of New Mexico enrolled in the study, my collaborator, and they were given a creative reasoning test. And we looked at whether there was differences between individuals with low and high creative reasoning scores. And we found that there was very high evidence of a difference using Bayes. We have a posterior probability, a probability that there's any difference in brain structure between these two groups. And it was quite high. And we also found that there was a lot of significant changes in connections in the frontal lobe. And so the bottom line was that there was evidence, and albeit a small study, that more creative individuals tend to have more connections across the hemisphere in the front of their brain. Okay. You could also use these tools to look at the effect of Alzheimer's disease, and not surprisingly, we find that Alzheimer's disease leads to many, many fewer connections in the brain for somebody with Alzheimer's disease. Okay, but here I want to focus more on these results here in the next couple slides. These are for the Human Connectome project. And so what are we doing here? So what we did is we looked at relationships between the brain connectome and each of very many different traits. And then we adjusted significantly for the fact that we're testing many, many times using kind of what's called a false discovery rate correction. And we're also reducing the brain to a set of scores, or real number scores. And so I could take a scan of anyone's brain in the room. The original brain connection structures is this really complicated mishmash. But I can take it and summarize it accurately using a vector of real numbers. Okay, like maybe 30 real numbers. Okay, and so then depending on those numbers, if I vary those numbers, it's going to vary this more complicated brain structure. And so what are these plots here? I'm just showing the kind of first 3 most significant numbers in that 30-dimensional vector. Okay, and so I'm plotting. I'm plotting these, and then I'm showing color-coded for people in this plot having a low or high value of an oral reading test, and in this plot having a low or high value to fluid intelligence, and in this plot having a low or high value of self-reported the most drinks you've ever had in your life in one day. Okay, and so what does this show? Well, it looks like even just taking a couple of brain principal components, we can have pretty good separation among the group of people who have a low value and a group of people who have a high value in this oral reading test. And that's only just using a couple of these scores. If we use more of them, we might be able to get more accurate. We can also see a little bit of separation in the substance use and in this fluid intelligence score. Okay, so what is this plot down here? So this plot down here is breaking the different traits we have data on into different blocks. And so this block right here is a cognitive trait. So that would be like oral reading test, fluid intelligence. They measured a whole bunch of different cognitive traits in this study. So this block right here is cognitive traits. Here's motor skills, data on substance use, information on psychiatric and life function, sensory information, emotion information, and we have a little bit on personality and a little bit on overall health, okay? Okay, and so now for every one of these different variables, we tested for relationships between that variable and the brain connection structure using this kind of cool method where we could summarize the brain structure with a bunch of scores, okay? And then the line here, we're doing a bunch of different tests, and so we need to kind of adjust the significance level of the test. If there's a bar above the dashed line, the upper dashed line is a higher level of significance. So higher is more significant, and the lower is still a pretty good significance. And so these are significantly related, even adjusting for the fact that we've done a bunch of tests. And so you can see if we look within the cognition traits, there's a whole bunch of them that are quite significantly related to brain structure, okay? And some of them are oral reading tests and fluid intelligence. And so some are quite significantly related. If we look in motor, they're very significantly related, almost all of them, except for this one. If we look in substance use, there's again a whole bunch that are significantly related. And so also in these other ones, in health, emotion, psychiatric life function. And so there's a ton going on in the data. There's a lot of signal in the data. Yes?
Were you able to replicate your studies? Did you get the same conclusions when you replicated?
Well, this is one study. We did look at when we had each one of these conclusions, we went back and looked at the literature in the area and saw whether there was other studies having related results. And so we didn't use that initially, we just looked at that. These are very early days in this field. And so certainly I would say that I wouldn't be looking at one of these results and then saying, oh, I'm going to completely change everything I do based on that. This is like observational data in an epidemiology study, and we're measuring associations. And so at this point, we need those other studies to kind of replicate these types of results. And so definitely keep that in mind as they go through. Yeah.
Can you elaborate on what dimensionality reduction techniques you use?
It's a type of, we came up with a modification of tensor PCA. And so it's principal components, but generalized to tensors, or semi-symmetric tensors, because we have this, and the current tensor principal component methods had a bunch of problems, and so we had to kind of modify them, modify the algorithm used for fitting them. Also, it's funny because if you're more technical, like if we use usual principal components, we have these orthogonality constraints for identifiability. If you use a tensor factorization and you put on orthogonality constraints, it's like incredibly restrictive and no data ever satisfy it. And so because tensors are totally different than matrices, and so you don't want to put down the same types of constraints, but you need to put down some sort of constraints. And so we tried to kind of carefully think through that issue. One of the other important things under the hood in the analysis is that you want to be careful about what you put in the tensor. So what's a tensor? Like a matrix is like some square array. So we'd have brain region by brain region. That would be like a matrix. Okay, well, we have a tensor because for each pair of brain regions, we measure multiple features characterizing the connection strength between those brain regions. And then we have brain region by feature, and that's like a block, like a Rubik's Cube or something, like an array or tensor. Okay? And now we have one of those for each person, and so we'd like to kind of characterize the variation among those Rubik's Cubes for each person's brain using kind of factorization in terms of simple parts. One of those parts is a numeric score of the loading of that component for that person. How much are they expressing that component? Yeah, and so, but you can see that like within substance use, for example, there's a whole bunch of them, and so it sort of adds to the evidence in some sense if we're doing kind of independent analyses for each of these traits, and then a whole bunch of them within a domain are all related. Okay. So this is kind of zooming in. So you could think that, well, we look at this, okay, well, one is like it's related significantly. Well, what does that mean? Can we actually map that back on the brain and see how the brain changes with the trait? Okay, and we can do that. And so the three plots here are describing how the brain connectome changes. We're adjusting for age and gender in the analyses. And I should mention that we did out of sample prediction, and then you could show the percentage of predictive improvement adding the connectome to demographic factors. It was surprisingly high, almost 10% with max strengths, and so that's quite significant given the amount of measurement error we have and everything. So. Okay, so let's zoom into here. The plot A, this is the brain. This plot is describing connections between different regions of the brain. So we have the two hemispheres of the brain. Okay, here's the front of the brain, and here's the back of the brain. Okay, and so the plot A is reading age-adjusted correlations 0.45, and so this is for a reading score. Okay? So what do we have? So what does a warm color mean? That means as the trait increases, we have more connections in those flagged regions. Okay? And so as your reading score increases, then you tend to have, that people with higher reading scores tend to have more connections in these regions. Cross hemisphere between these, this part and this part, and then within a hemisphere between these parts. Okay, okay? Does that make sense? So those are significantly increased. So now let's look at, so this is a good trait. And so we expect for good traits to see more connections if you have a higher value of the trait. And that's what we see. There's no blue ones at all. We could see blue ones, but every one we saw was kind of orange or red, suggesting that they're all in the direction of more connections, a better reading score. This one is actually our most significant predictor, which is I would call it a binge drinking predictor. And so if you were in a very high category of this predictor, you would have self-reported that the max drinks you've ever had in a day is 21 or more. So you've had 21 or more drinks in a day, which is quite a bit. by my standards anyway. And so this shows, now these are all blue. Why is that? Because as the max drinks increases from a low value to somebody who only drinks like max drinks maybe 4, 5, 6, 8 maybe, to somebody with like in this 21 or more category. So as that happens, the people in the high category, they tend to have fewer connections between these brain regions, okay? The ones marked in blue. Okay? And this was very significant, actually, very, very significant. In fact, what was our classification rate? What was this? So if we group people into low value of in the binge drinking trait and high value, and we just tried to predict based on their brain structure whether they had a history of binge drinking or not, our classification accuracy out of sample was 81%, okay, which is pretty high. Maybe it's confounded. This is just observational data. This is not showing at all that binge drinking causes brain damage or something like that, but it's certainly disturbingly suggestive. The other thing I found really interesting was we had a current use of marijuana variable. So look at how similar these are. I mean, there's no reason that they should have been similar. It's very similar. It's also significant. We didn't have as good an out-of-sample classification rate. Wasn't very good, but it was statistically significant, and it picked up a very similar set of connections being less among the people who smoke pot. Okay. So reading increases connectivity, substance use decreases connectivity. Okay, and so we can also kind of zoom in and examine brain network differences on the individual level. These are kind of population averages, and so we can kind of pull off people, individual people, and then see how their individual brain was kind of impacted. And that's what we kind of did here. And it kind of shows this is a reading, and this is the max drinks trait. And so for this individual, they didn't have that other part. They just had this kind of front part that was lower, and then it'll kind of show where in the brain these occur. Yeah. And so let's focus on significant cross-hemisphere connections here, and then we can look at differences between subjects with high or low reading scores, for example, and then you can kind of find these differences. Okay. So I don't want to go into that in too much detail. I think mainly the story was here, and we're out of time. And so let me just wrap up and say that there's some conclusions, I think. There's a lot of interesting conclusions and areas for future work and kind of implications, kind of societal implications. I'd say an individual's brain structure is clearly significantly associated with different traits of that individual. It would be very interesting to run a kind of more of a study or a randomized study for different treatments or something, or a way to kind of establish more of the causal effect, or run a longitudinal study that get better exposure assessment. That's quite important. But now the key part is that we kind of have the technical or the data science tools to figure out these relationships, which has been quite a recent development. Like before, a couple years ago, you could just get a bunch of noise in trying to reconstruct these brain connectomes. And people have heard of fMRI. This is not fMRI data. There's a lot more signal in the structural connectome data if you do it properly. fMRI data is very, very noisy and it doesn't replicate very well. So the next steps is we wanted to go beyond flagging associations to kind of acting on this knowledge. Can we actually show some factor X leads to a particular change in the brain connectome? It's sort of what you call in statistics is causal inference. It causes. If we didn't have that factor, we wouldn't have that change in the brain. Can we use it to improve diagnosis of mental health conditions? I would love to get a bunch of data where we had people with different mental health problems and these connectomes and kind of work from there. Can we design training programs for the brain for both healthy and pathological individuals? And I have an actual grant from the Army to kind of look at some of these things. And so we would like to be able to, somebody comes in, you can imagine, well, if we look at their brain, we might be able to kind of figure out what they're good at. put them in that training program for that, and then we could kind of even look at the brain after that training program and see what impact it has. And so that's pretty interesting, I think. There's a lot of really, related to that, there's a lot of really interesting ethical or policy implications. So if you had your brain scan, if it looks good, should you include that along with your CV and your job applications? Does one's brain scan impact insurance coverage if that gets out? If you're, for now, it's funny because, I shouldn't say that because relative to genomics, it tends to be very hard to get, if you're working on data science and you want to work on some interesting data. It's nice for the data to be publicly accessible because then lots of people can work on the data and improve methods and everything and come up with lots of data science tools. It also will allow you to validate the methods because if everyone can look at the data and they're like, well, you screwed up, you like miscoded everything and like your method sucks, they can tell that because the data is publicly available and then some other data scientist can replicate the results. But for a lot of medical data, you can't make the data publicly available because there's these considerations, concerns, so certainly for genomics. But for now, a lot of the brain scan data is publicly available. And so that it will be interesting if it contains a lot of information relevant to insurance coverage, et cetera, then it might not be as publicly available. And then the science kind of slows down dramatically as it becomes not publicly available. What if we can accurately flag individuals at high risk for pedophilia, violence? I have a collaborator who's really interested in violence. She went into a prison and took brain scans of the prisoners, and now she has this kind of whole data, which is really cool, where the people will play adversarial games against each other, and we take videos of their face, and we try to infer their emotions and things. An important point, I think, is that the brain connectome is not fixed and predestined even in adults. I would say that's true based on our studies, other people's studies. It's pretty plastic, the brain. And so you could see that when you wounded it by putting in this electrode array, all these cells kind of come in. But even like an adult, you go to some training program or something, your brain, or you sit around and play video games all the time, your brain's changing. Yeah. And that's that point there. And here's just a bunch of references, and I'll just stop there.